This is the second video for the Genetic Regulation Unit. In this video, we will discuss the variety of ways that eukaryotes are able to regulate the expression of their genes. Once the nucleosomes have been removed from the promoter region of a gene, there are multiple levels of regulation that can occur. The first level is initiation of transcription. In order for transcription to occur, general transcription factors first bind to the promoter to recruit RNA polymerase. This process is regulated by controlling the recruitment of the general transcription factors to the promoter region of DNA. This is done by regulatory transcription factors. Certain types of regulatory transcription factors, called activator proteins, bind to sites in DNA called enhancer sequences. These activator proteins recruit the general transcription factors to the promoter by binding to them. Once the general transcription factors have bound, they then recruit the RNA polymerase and transcription can occur. There are another set of regulatory transcription factors called repressor proteins which bind to silencer regions of the DNA. These repressor proteins act in an opposite manner. They prevent general transcription factors from binding, thus preventing transcription. Genes tend to be associated with multiple enhancer and silencer sequences, which can be close to the gene or further upstream on the DNA. This means that transcription of a gene depends on the presence of a particular combination of regulatory transcription factors. This process is called combinatorial control, and transcription will only take place when all the regulatory transcription factors are present and working together. RNA processing is also important in gene regulation. A particular type of RNA processing regulation is called alternative splicing. Remember from previous material that splicing involves removing the introns from a primary transcript to produce the messenger RNA for translation. Alternative splicing is a regulatory process where introns can be removed in different patterns, resulting in different messenger RNA sequences and different translated proteins. In this example, we see a region of primary transcript with the 5 exons labeled 9 to 13 here. In liver cells, when this gene is transcribed, all 4 introns are removed and the 5 exons are joined together. However, in skeletal muscle, splicing of the second and third intron is completed as if they were one large intron. Exon 10 is joined to exon 12. The loss of exon 11 means that this messenger RNA will be translated into a slightly different protein than the messenger RNA produced in a liver cell. This protein is the insulin receptor, which is present on the outside of cells. In liver cells, this receptor has a low affinity for insulin, so it will only bind when concentrations are very high. But the version produced in skeletal muscle is a high affinity version, so it can bind insulin even at low levels in the body. This difference allows muscle cells to better absorb glucose, which they need for cellular respiration. Small regulatory RNAs are a category of regulatory molecules made up of microRNAs and small interfering RNAs, which are able to regulate gene expression by either blocking translation of other messenger RNA molecules or causing the degradation of messenger RNA molecules. Small regulatory mRNAs are transcribed from genes like other protein encoding genes. However, RNA itself is the functional product. They are not translated. The RNA is transported out of the cell either as a double-stranded RNA molecule or a hairpin loop. The RNA is then cleaved into a small RNA fragment. And a single strand of that fragment is incorporated as part of the RNA-induced silencing complex, or RISC. The single-strand RNA will form complementary base pairs with various messenger RNA molecules, causing them to either be degraded if the regulatory RNA exactly matches the messenger RNA, or inhibiting translation if there are some mismatches between the sequence of the regulatory RNA and the messenger RNA. Gene regulation can also occur at the level of the protein, either during its production through translation of the messenger RNA or after production through modification of protein structures called post-translational modifications. Translation can be regulated at the level of initiation, where regulatory proteins can prevent ribosome assembly on the 5' cap or block translation through the 5' UTR region. After production, post-translational modifications by additions of phosphates, methyl groups, or sugar molecules can control the activity of proteins. For example, digestive proteases that are secreted from intestinal cells into the intestine in order to digest your food would be very dangerous if they were active inside your cells themselves while they were being translated. These proteins are modified to keep them inactive until they get to their proper location.